Good morning, good afternoon, and good evening, everyone. Thank you for joining us today for today's session. I know that people are calling from around the world, so we do appreciate you taking time from your busy schedule to join us today. My name is Sharif Hanala, and I am a product manager here with the Bio-Rad Process Chromatography Group, and I will be moderating today's session. Today's webinar is entitled Enabling Process and Intensification Using Modern Chromatography Media. It's a great complement to the webinar we held last week where we discussed more specifically using mixed mode selectivity for specific unit operations. Today we will take a step back and perhaps look at processes holistically and highlight ways that chromatographers as process developers could significantly optimize processes by making them more efficient just by doing what we do best, which is either using new novel mixed modes or using established functionalities a little more creatively. Today's session is going to last about an hour with a talk time of approximately 35 to 40 minutes. We hope to generate a lot of questions from the audience, so we do have about 20 minutes at the end to discuss questions. Those of you who've joined us before, you'll notice that there is a Q&A tab at the bottom. It's at the bottom of your console, and you could just type your question in there, and I will read them out to the speaker as we proceed, or perhaps at the end. And if you have any technical problems, you could also enter them in there. At this point, I have the distinct pleasure and honor to introduce our speaker today, Dr. Carsten Voss, who is a key member of our group here at Bio-Rad. He's calling in today from Germany. Dr. Voss has studied chemistry at Bielefeld University, and he obtained his PhD from there, and I also believe he has maintained a lectureship there in biochemical or bioengineering. For the last couple of years, Dr. Voss has worked with a variety of groups, CMOs, and innovator companies, as well as a range of customers of Bio-Rad on purification problems. And he will be walking us through the presentation that I described a little earlier today. So at this point, I'll just give control over to Carsten and let him take it from there.
Yes. Sharif, thank you very much for the kind introduction, and welcome to everyone attending these webcast here. It is my pleasure to tell you about Bio-Rad's process chromatography solutions. As you can see here on this first slide, Bio-Rad has been in this business for around 50 years now. And for people who actually don't know it, Bio-Rad really started as a chromatography company around 60 years ago now. And what I want to highlight today are the new resins in our development that are mainly being the Nuvia SNQ high-capacity ion exchangers. It will be the Nuvia HRS, which have as a high-resolution cation exchanger. We have mixed mode resin next to the hydroxyapatites. We have a new cation exchange hydrophobic interaction resin, that's the Nuvia C prime. And these are the resins that will be focused on during this presentation here. The question you might ask is, when I'm developing a process, I know my chromatography resins, I know how they work, so why should I change? A reason to change is that the healthcare industry that you are developing for and that you are producing for is also a fast evolving landscape there. So patients have been cost unaware before that, now they turn into consumers who are value conscious. Historically, we've been looking into drugs that were one size that should fit all the patients. Now we have a tendency to look into more personalized medicines. Biologics have been used only in the developed world. Now they get to be applied everywhere and also produced everywhere because processes can get more economically feasible. Drugs, diagnostics, and devices turn into one unit in one operation that could be called theranostics. Also, researchers not just look into age-related diseases, but looking at aging itself. That's also an interesting development. that has been in discussion recently. So aging should be considered as a biochemical process, of course, and this can also be manipulated, probably not be treated at all, but the aging process itself can be mitigated, and also the associated illnesses and sicknesses that we have. And finally, of course, researchers not look at now just treating the sickness and curing the symptoms, but also finding methods to really prevent these sicknesses. Another reason why you should go and look into new methods for developing process chromatography is also that these healthcare trends, of course, have impact on biomanufacturing. And you will observe several trends in the recent years. Of course, you have novel therapeutics that are coming up that meet new approaches in biomanufacturing. You also have a tendency to have multi-product facility and not just facilities dedicated to the production of a single therapeutic drug. Also have flexible and continuous manufacturing. Continuous manufacturing is also in discussion during the recent years. Healthcare economics in a healthcare system in different countries, as well as strong competition, also lead us to find lower cost processes also to be transferable due to the globalization of our market there. And of course, you will also see that new resins and membrane functionalities come up. And this is where Bio-Rad process chromatography comes in. That's our core business to produce these new resins. And we want to encourage you to use our expertise where it really matters there in these new trends and new challenges. So when we look at novel therapeutics, of course, we have unique purification challenges, like when you look at new antibody-related drugs, like antibody-drug conjugates or multispecific antibodies, you will see that you have, for example, molecules to separate that they are structurally close, very close related there, and that's a new challenge there, where not a classical platform applies. This also goes for other new therapeutic proteins, but when you look on the established proteins, you of course require very productive approaches, because the margin you get out of selling these products drops. The competition comes up with evolving biosimilars on these established proteins, and that's why you look into a more productive approach there. And challenges for those molecules can be, for example, that you have restricted stability windows. Some proteins need a high activity, correct folding, or cofactor to evolve the full therapeutic effect. And on the other hand, of course, the economics must work after all. So you need cost-effective and a robust process to generate a viable therapeutic there. And what I want to do today is to highlight some cases of how these challenges can be faced with new chromatographic resins. And I want to highlight on the mixed mode selectivity and the high resolution ion exchanges and high capacity resins. So what I will do today is present 6 cases where such functionalities are applied. The first case I want to address is an enzyme purification process that will be modernized and simplified there. And this enzyme purification is kind of a role model for several non-antibody early phase candidate therapeutic proteins there. And we've been looking at a process with a large number of steps, including precipitations and a very costly chromatographic purification steps there. And we had the goal to modernize and simplify this process. And In this slide, you can see in the top the current process, which is very complex and results in a very low activity recovery. So for an enzyme, it's not just important to retain a lot of material, but also keep it in an active state. And that's why we defined here the aim to maintain the full activity recovery. And the first steps here are two: precipitation to concentrate the target protein. These precipitation steps that are difficult to scale up were followed by ion exchange combined with expensive affinity step. Third chromatographic step here is a hydroxyapatite polishing step, which is succeeded by a second anion exchange step, and that is followed by size exclusion. Size exclusion here is, of course, with good resolution power for size, but difficult to scale up there. And as a final step, the process uses affinity by an immobilized cofactor on agarose there. And as you can see, the overall activity recovery is reduced to 9.5% there. And we were looking at a very simpler process with fewer steps and higher activity recovery. And what we really did is to look into one of our early developed process chromatography resins. That's analytical grade anion exchanges, the AG1XA. And this is an interesting resin to be used in flow-through steps, for example, to remove DNA, and it can be put in front of a chromatographic purification scheme to be used in negative chromatography. The second purification step was an ion exchange hydrophobic interaction mixed mode chromatography, which combined these both interaction and could replace the different affinity and ion exchange modifications here. Finally, polishing was achieved with the ceramic hydroxyapatite, CHT called here, and as you can see, a very high activity recovery is retained there due to the efficient purification and the purity of the sample we have achieved. You will have an activity recovery that is above 100%, so that's not surprisingly if you look at enzyme activities there. But what is important is that the number of overall purification steps was completely reduced. This is not only important for the economics there with respect to chromatography resin, but also, of course, when you look at your buffer consumption there. Good. The second case that is to be presented was the purification of a therapeutic protein. This development was in a very early phase there, and the current process had the trouble that the product was degraded during purification process. And we were looking into that process to be optimized. And on the top you can see the current process starts by harvesting at neutral pH. You do a solid liquid separation by tangential flow filtration and adjust the pH to 8. And in negative chromatography mode by weak anion exchanger, the product is kept in the flow through. And secondly, ion exchange mix mode is applied for positive chromatography, and the material is polished with a CHT as a final step. And as you can see, the overall process yield in this purification was below 3 mix per milligrams per liter there. That is less than 10% of the process yield that we achieved with our development there. The trouble that the customer observed here with this process was not only that of course you have a loss recovery losses during all your chromatography steps. That wasn't the worst thing. But the worst thing was that we definitely observed proteolytic degradation of the target protein during this process. And of course, this was difficult to come by. This was observable by monitoring, simply monitoring the fractions in this process by the SDS pages there. And we've been modifying the process, bringing in high-capacity ion exchangers and mixed mode resins. And what we did, of course, was starting with a harvest at pH 7 there, but then, of course, for reducing the proteolytic activities, work at lower temperature there. Remove first impurities with a Nuvia Q that got a high binding capacity. For that purpose, keep the product in the flow-through. We adjusted the pH to a lower value and exchanged it by tangential flow filtration. It was also supplemented in the final process by moderate concentration of EDTA to complex metals that work as a cofactor for the proteases that was at work here. And the main purification steps here was the cation exchange mixed mode resin, which could purify the product, and that was finally polished at almost neutral pH by CHT as already outlined in the original process there. And by modifying this protocol using mixed mode, adding EDTA there, and then working at an acidic pH with the Nuvia C prime, we were capable of increasing the process yield by a factor of 10. So that was a tremendous result for our customers that asked for our support here. The third case is early stage therapeutic enzyme process, looking at different isoforms in this process of this enzyme that has to be resolved there by chromatography. And for that case, we used a dedicated novel resin that's been in development, being an anion exchange hydrophobic interaction resin. And as you can see on this slide here, you've got chromatogram with fractions 1 to 18. You see main peaks in the flow through. These main peaks definitely constitute impurities that have been removed, as can be outlined in the SDS page on the right there. And 2 elution steps were capable of eluding different enzyme pools there, which were able to identify that these were 2 enzyme isoforms with different specificity there. So this was done with an anion exchange hydrophobic interaction mixed mode resin. Case number 4 was a customer case where the aim was to purify a highly glycosylated viral antigen that was in an accelerated development program, and our customer had the trouble that he's been using a lot of single mode chromatography. Definitely everything that's available from ion exchange, hydrophobic interaction, affinities for highly glycosylated molecules, and nothing really gave the desired purity. He was looking into size exclusion. It was working well, but of course you have again the scalability issue. And so we proposed to test the Nuvia C prime for that as a novel ion exchange HIC resin, and we were taking a look at this. And when you look at Nuvia C prime, you have definitely 2 parameters to work with. That's the conductivity, of course, modifying the cation exchange interaction mainly, but also you have a weak CM group and a hydrophobic interaction there, and of course conductivity and pH there go hand in hand there. And what we could do here was to purify the protein by binding it at phosphate concentration, citrate buffer with 250 millimolar concentration at a low pH there. So under these conditions, both the target protein and impurities bound to the resins, and we were applying a gradient to a higher pH. At this higher pH, you have electrostatic repulsions coming up, and so you've got the CM group on the ligand as well as the proteins getting more negatively charged, and by this kind of hydrophobic charge induction chromatography, you get removal of impurities, as you can see here in the SDS page, and the lanes with the orange gradient 1, impurities go off. By holding it at a high concentration of phosphate citrate at pH 8, you can see here in this holding step even more impurities were eluded there. And secondly, as showed in the gradient 2, we used a decreasing conductivity gradient, keeping the pH constant here, going to 0.05 molar phosphate and citrate. And in this case, we could show that by mitigating the hydrophobic interaction there, the protein was nicely eluded. So this is a very interesting example how you really can exploit both parameters to really capture, bind your target protein, get rid of the bound impurities, and then modify another parameter and elute your protein off the column. Case number 5 is looking onto IgM antibody. So these IgMs are used in diagnostic applications, but also find a way into therapeutic treatment there. And for these IgM, really no platform process is available. What we have been asked here is to look into a dedicated separation challenge. The feed material contained the IgM as well as the very large content of serum albumin here as an impurity. And in principle, with single mode interaction, you would have the opportunity to anion exchange to bind both the IgM and the serum albumin at a higher pH there. On the other hand, you could use probably cation exchange, but since the PIs are very closely related to each other, that might be a little bit of a difficulty to get a very nice separation. So both single mode interactions there were mainly ruled out, and we were looking at the Nuvia C prime as a cation exchanged mixed mode resin. And as you can see in this chromatogram that is done with the C prime resin, you can see that impurities, mainly the BSA here, are kept in the flow through. The IgM is bound to the column and can be eluted in a step elution with increased salt concentration. And the result of the chromatography step here is nicely illustrated in the SDS page to the right, where you can see the C prime eluate in the right lane with no impurity at all. Case number 6 goes to MAP process, where a customer was seeking quite a robust alternative for aggregate removal, which is a common issue for monoclonal purification. They wanted to explore such an alternative to be introduced into their platform, and they had an ion exchanged Higg mixed mode, which was not their ideal solution, and they had to bring in tight control to work with that process. And we were looking into offering a solution and tuned cation exchanger by modifying the bead size and the pore structure. And this customer requirement and development led to a resin that is based on the ionosphere rapid as material. The chemistry is virtually the same, but as outlined before, the bead size has been reduced to a mean size of 50 microns, and the pore size has been optimized, and this resin is now commercially available from Bio-Rad. It's called the Nuvia HRS, high resolution chromatography resin, and as you can see, it's got more effective resolution power in comparison to Nuvia S or the Ionosphere Rapid S by the smaller particle size. And this is outlined in this slide by the separation of some model proteins. The application of the customer was, of course, to use it for aggregate removal, and this was done here. An example, antibody purification protocol, where you could see that sodium chloride gradient was applied in a pH 5 acetate buffer there. And as you can see in the linear gradient, you get good separation of monomer from the aggregates. You have fractions here that's been analyzed by SEC, and the size exclusion profile from these eluted fractions is shown down here in the right corner. And as you can see, the fractions 17, 18, and 19 representing the main peak, which is to be the monomer, was confirmed by SEC that this is the monomeric fractions. Even if you turn in the right shoulder of the main peak, this is going to be also mainly containing monomer, just increasing the aggregate content slightly. And as you can see, the later eluting fractions from the cation exchanger mainly constitute the aggregates as confirmed by SEC there. So that's a good example of exploiting high resolution materials for that purpose. We've been looking into different applications now with customers for that material, exploiting this high resolution powers there, especially interesting for separating structurally closely related molecules, also target proteins with close PIs, where we have successful results to present. So, in summary, I've been able now to show you a few cases with new selectivities and high resolution chromatography media, and there is, of course, more to come during the rest of this year, where we are going to present on conferences and on fairs there. And today you've seen insight into process where we have been able to improve yield and maintain activity by reducing proteolytic cleavage in an enzyme purification process. Also, these high resolution and new selectivity resins offer you to reduce the number of process steps and therefore overall cost of the process. And what you should also take into account is that you have to the opportunity to minimize your feed manipulation processes. For example, cation exchange mixed mode resins are higher salt tolerant than the classical cation exchanges, and so you probably don't have to look into opportunities or ways to manipulate your feed for subsequent purification schemes. Finally, we also had the achievement of separations here with new selectivities, where all classical interactions didn't give satisfactory results, so they were out of reach with traditional approaches there. As a conclusion, I would also like to outline an opportunity here for you to work with Bio-Rad there. So I particularly like this slide because it's also outlining my work at Bio-Rad. What we do in process chromatography is, of course, that our group develops new products to be used as resins there. You, as a customer, are facing us with new separation challenges, new molecules, and my group from the application side is really what makes your challenges and our new products interact there. So we are the glue, we are the wheel that is in between there, and we all together, all three groups together, make the innovation really work for us. And that's how things should going. And for today, I am now happy to take your questions that you have and look forward for an interesting discussion with you. Thank you very much for your attention.
Thank you very much, Carsten. Unfortunately, we can't hear the crowd clapping, but I'm sure they are. I'm sure they are. I certainly am. Thank you for presenting these cases. I know that there's a lot of detail that goes into developing this kind of work, and we'd be happy to share some of that with the audience if they have questions. They could reach out to you directly or to anyone from the Bio-Rad chromatography group. We do have a few questions from the audience that I'd like to maybe read out to you, and perhaps get your comments on that. Just for everyone, before we get into the Q&A, just to let everybody know that this webinar is going to be archived and available for you to see or share with your colleagues, probably within 24 hours on the On24 platform, and it will be available there for a number of months afterwards. And again, if you have any specific questions regarding any of the products or applications, please reach out to Carsten, and I'm sure he'll be able to put you in touch with the right group. So the first question that I have that I've received is, I believe you talked about several mixed modes, different kinds of mixed modes, and the question that I have here is, how do I select the right mixed mode for my application, be it something like a CHT or an anionic, cationic, or what have you. So maybe you can have a comment on that.
Yeah. So what I want to refer to when looking at mixed mode, I would say there is no really right or wrong there. Selection of mixed mode, of course, depends on your target proteins, the impurities you have, and your separation challenge. So that's always the same it goes. And when looking at like Nuvia C prime, you would virtually look into developing methods for such a mixed mode resin as you would develop a method for a classical cation exchanger there. You know that you have protein that is capable of binding to a cation exchanger. You can either modify their interaction with the conductivities and the pH there, and it really depends on the nature of the protein. When you look at mixed mode, you have two interactions. What interactions will dominate? It is really not predictable how to select that, and that is why we're offering tools to support our customers on developing purification schemes with mixed mode resins. For example, guidelines for DOE with microwell plates or working with robotic columns there. And that is a way to find a good solution with a mixed mode resins for your separation challenge. Another opportunity is, of course, look into opportunities that come along with mixed mode there. If you want to have binding and higher conductivities, this can be exploited by a resin like Nuvia C prime because of the additional hydrophobic moieties. That's also things you have to take into account, and in this case you can use the mixed mode resins to probably work as a combined two orthogonal steps there. So, but finalize this answer, Sharif, here is even if you've been looking at mixed mode before, you've been using C prime or another resin for a definite separation of a protein, try and screen it for another protein again, because it can behave quite differently. There is no good or bad, no right or wrong. All the mixed mode resins are just different because of the different chemistries that we have.
Okay, well, that's okay. Thanks for that answer. We could also provide some assistance if of course, if needed. So the next question, I think I'll take it, and it's a question that I've received before, and I think the question is how expensive are mixed mode resins? And the way I'd like to answer that question is really rather than talk about the price or expense, maybe discuss the value. We work really hard to develop resins that unlock value in the process. So if there's a way for us to allow you to do a purification that would have been otherwise impossible before, or one that would require several columns or extreme process conditions with a mixed mode, I think ultimately the price becomes secondary, because you are doing something that you couldn't do before, or you're saving a lot of time and money and developing a more robust process. But to address the question directly, mixed modes are generally more expensive, of course, than ion exchangers, because they are more complicated to make. The ligands are proprietary, so those have to be synthesized and developed. But they're not anywhere near as expensive as affinity resins or any kind of specialty affinity resins that are on the market. So I would say that they're not over expensive overall, and if you know how to use them properly, ultimately you end up saving quite a bit of money and increasing your process quality. So I hope that's a good answer for people in the audience. And if there's any specifics, or if you want to discuss that, pricing or to look at acquiring some of these resins, we'd be happy to speak with you individually. So the next question I think is for you, Carsten, and it is regarding the difference between something like CHT. So the question specifically is, how does CHT differ from Nuvia C prime or other mixed modes that are on the market?
Yeah, that's a good question. The difference is quite easy to identify. The first is, of course, obvious. It's the chemistry. When looking at the chemistry, you will realize that the mixed mode interactions are different. CHT is a cation exchange. IMAC interactions, where you have the M groups of the proteins and phosphate that can interact with the calcium moieties, and you've got, of course, the classical cation exchange that is working. C prime is mainly a cation exchange and a hydrophobic interaction that is working in the mixed mode operation there. The second difference is that, of course, CHT is a rigid ceramic material made from the calcium phosphate mineral hydroxyapatite there, while C prime is a mixed mode ligand that is attached to a synthetic polymeric bead. And this polymeric bead, in contrast to CHT, is compressible. And when looking at the differences, I also want to outline what they both have in common. When I look at method development, for both resins, method development is really straightforward and not really rocket science. In the CHT case, as outlined before, you manipulate the cation exchange and the IMAC interactions mainly by working with phosphate and sodium chloride concentrations. And in case of Nuvia C prime, it is working with conductivity and the pH value of your buffer system there. So that's different and what they have in common.
Okay, cool. So they're distinctly different materials. Okay, the next question says here, I have a process that will probably have 5 chromatography steps, and I'm thinking of maybe using mixed mode to reduce those number of steps. Where to begin? Where to begin thinking about inserting a mixed mode step? The beginning, the middle. I assume the person is trying to just basically find a way to reduce the number of chromatography steps, and they're struggling of just how to start this process.
Yeah, very difficult to give you a really generic recommendation on how to proceed. But when you look at purification process development, you usually have combinations of orthogonal techniques there. That is going to be affinities and ion exchanges and hydrophobic interactions there. And from my perspective, mixed mode is an interaction that is somewhere between the single mode interactions and very specific interactions like affinity. So since it's being somewhere in between and including two orthogonal techniques there, I would look at trying to substitute two orthogonal steps by implementing just one single mixed mode step there. And secondly, you can also look where you can exploit the higher fault tolerant to use the ion exchange functionalities under different conditions where it's applicable. But that's a first goal really to go. Otherwise, sometimes you, of course, have to see every protein is different, and it's not so easy to predict that for their behavior in mixed mode. So we've been looking into several different proteins and how they behave there, and we've identified proteins that work with the Nuvia C prime more in a cation exchange interactions. Other have strong hydrophobic interactions there that is showing that they behave in a mixed mode order with Nuvia C prime there. So it is depending on your protein, really, how the mixed mode efficiently works in this process.
Okay, so I think in a nutshell, perhaps, if you know something about the protein, that certainly helps, and then maybe consider inserting replacing an ion exchanger, let's say after a dilution step, and avoid that dilution, or maybe if you have a hic in your process, you may want to start there and start thinking of the mixed mode as a hic, and then see what you can do with it electrostatically. So, perhaps these are maybe two places where you can consider. Of course, there is also the option of using it as a capture resin. The person hasn't specified; they just said protein. So, obviously, the more we know about the protein, the better, of course.
Exactly. That gives you the most decent clues there. If you have detailed information on your protein that go beyond theoretical PI there, it is always helpful. Do also process analytics. We have excellent tools there. We could really showcase in these examples that I've been showing here that also in process data in the development steps really helps. So we were able to identify the proteolytic degradation, where it was working, under which condition it was working, by simply monitoring it with SDS page. It hasn't to be really complicated. Sometimes it's just taking the time, observing, and you get the information you need to efficiently develop your process.
Okay, so I think we have one last question, and again, we still have a few minutes, so if you have other questions, please post them. I'll take this one. It's regarding GMP processes using mixed mode functionalities, and the person is wondering if there are any or many, and yes, there are. For a case of CHT, for instance, there are several dozen processes, and many of them are commercial marketed drugs that have a CHT polish or a CHT intermediate step in their process. As for Nuvia C prime, the product has been on the market for approximately almost 2 years, and we do have several phase ones or materials that are entering phase one that use C prime, mostly on the sort of therapeutic protein side. So overall, and if I look at some of the other competitor resins, I believe some of those are also used. So overall, as a class of resins, the mixed mode functionalities are becoming very well accepted and included in GMP processes that are FDA approved. So if you are considering sort of this class of resins, I don't think you should worry about any kind of regulatory impact or whether these resins are going to remain on the market, because it has already become a very established class that is available to you as a tool. So that would be my comment on that. I believe we don't have any further questions. Let me just do another refresh. Yeah, I think we're done for today in terms of questions. So are there any sort of parting remarks that you want to add, or something that you may want to correct in what I said, or anything like that?
Thank you, Sharif. I think everything was fine from your side. No corrections from me.
Okay, very good. Yep. Okay.
I want to thank the audience for paying attention, and if further questions come up, you see my contact details on the final slide here. Just don't hesitate to come up and send me an email there. I'm fine. I'm happy to answer. Happy to go into discussion with you.
Okay, I got another question. Just when we thought we were done, I have a question from the audience. Let me just see here if I could just get that to you. Essentially, it says, can affinity resin such as like a nickel iMac be engineered? Hang on. Let me just send that to you so you can address it directly. But it says, can a nickel resin be engineered into a mixed mode resin to reach better purity in one column? Let me take a shot at it. I'm not sure what engineered means. I mean, do you mean that this resin could be modified in some way or used differently? So perhaps the questioner could maybe add a clarification. But what do you think? I sent it to you, Carsten. What's your take on this question?
Yeah. So what we're looking here at is something like a mixed mode that is contains an iMac functionality and probably also another functionality. Yeah, I think in principle we wouldn't have a problem to engineer such a resin there. You could put that probably the functionality into one ligand there. Also probably add two ligands to the resin there. In principle, it's possible. There is nothing that the chemists in our labs almost can't do there. The question really is if you want to have a classical iMac functionality on process resin for pharmaceutical purification there, because iMac is a very nice tool to work for lab scale purification, to work with a histech or proteins that naturally have a strong histidine content there. But of course you're facing other problems like working with heavy metals here and metal leakage from the column there, and this raises the question if you really want to apply such a resin at a process scale. But generally the question can be answered to be yes, it is possible to engineer such a mixed mode resin by our chemists, and better purity in one column also depends on your application. But I think, of course, it will have an effect, same as interactions like ion exchange and hydrophobic interaction have.
Yeah. Okay. Yeah. I mean, it's a valid question. Obviously, there's a reason why nickel iMac exists, and that it stands alone as an effective tool. So I guess we wouldn't want to compromise that by adding other functionalities. But you've also raised some very a comment that I want to add to this discussion of how to invent or make a new mixed mode. I mean, there's a variety of ways we can do this. The question is, for us, are we bringing a new tool to the market? Are we bringing a resin that has a unique selectivity to the market? If there's something you could already do using the tools that are available, we don't feel the need to create a new resin. A new resin is only sort of launched when we feel that there is a selectivity gap that we can address, or perhaps an operational gap that we can address. So our resin development program here at Bio-Rad, and I'm sure other vendors, is always looking to bring new tools that allow you to do something differently, as opposed to just slightly modify something that is already available and effective. So that's sort of a general philosophy on sort of resin development, attitude from a vendor's perspective. Okay, so I think that's all we have now. there's always an opportunity to ask more questions and ask for more data. I'd like to maybe just let everyone go about their day or evening and thank our speaker very much today for his insight. And as always, whether you are in Asia or anywhere around the world, in Europe or North America, we do have experts that are within your reach. All you got to do is sort of reach out to your Bio-Rad chromatography specialist or to Bio-Rad in general or Carsten or myself, and we'd be happy to answer your questions or see if we can directly assist you with your purification. On that note, I'd like to thank the organizers and everyone who attended, and we look forward to another webinar series as we sort of tackle the industry challenges together. Thank you again for your time, and hope to see you again and talk to you again in other venues. Have a good day.